Hi, welcome to Critical Thinking for Ecosystem Science. I'm Dr. Tom Stolgren. This is Lecture 4, Looking for Truth in Personal Experience. It loosely follows Schick and Vaughn's Chapter 5. Here are some basic concepts that they covered in the textbook. Just because something seems or feels or appears real doesn't mean that it is. When evaluating a claim, look for disconfirming as well as confirming evidence. When evaluating a claim, look at all the relevant evidence, not just the psychologically available evidence. Personal experience alone generally cannot establish the effectiveness of a treatment beyond a reasonable doubt. And case studies alone generally cannot establish the effectiveness of a treatment beyond a reasonable doubt. So let's investigate these just a little more closely. There's a Twitter site dedicated to looking at objects that appear to be faces, either human faces or animal faces. And we make them from everything, from a cappuccino cup to a loaf of bread to a face in cheese. We find images in things that we look at. It's perfectly human. And the mind can play tricks on us, we know, from a whole group of optical illusions. So you've seen these since you were a child, probably. Are there 3 bars or are there 2 bars on this one diagram? It depends which side of the diagram you're looking at. And of course, there are hoaxes. There are people that claim to show flying saucers by holding a string, and Photoshop has made this a very easy thing to do, to trick your eyes into believing that you may be seeing a flying saucer over your city. Just because something seems, feels, or appears real doesn't mean it is. And now in this study of extrasensory perception, the investigator would be staring at a card and you'd be across the table trying to guess which image is on the card. The circle, the plus, the squiggly lines, the black square, or the star. And for the most part, these studies have shown that, well, ESP, if it's there, is very rare. Where they have found evidence of someone being able to interpret those cards, they've also found some fraud. Sometimes the cards could be seen in the investigator's glasses or even in their pupils. Police lineups, eyewitness lineups, select an innocent person 18% of the time. We think we saw that person earlier, but 18% of the time we're wrong? That's pretty high. Why is it that a lot of the pictures of Bigfoot are fuzzy? I mean, people go out and they take clear pictures of trees and meadows and roads all the time, and yet whenever they try to take a picture of Bigfoot, it's fuzzy. Psychics, well, they cover this pretty well in the book. Every single psychic that has been tested scientifically has turned out to be some sort of a fraud. Why? Well, predicting the future to any great detail is impossible. It's just impossible. We're not that good. We can't see into the future, no matter what we use. Superstitions. They seem harmless enough. I believed in them as a child. A 4-leaf clover would bring you luck. A horseshoe would bring you luck. The number 13 would not. Wearing garlic might prevent, of course, werewolves. Crossing a black cat's trail would be bad luck. Breaking a mirror would give you 7 years of bad luck. And Friday the 13th, worry about those. But these superstitions, although they seem harmless, are a way to tell you that it's okay to just believe things without testing. And so I look at them as just a little dangerous, really. That's me. You might have your own opinion. It's all right. We can be different. Let's talk about the placebo effect. Now, that's where you wander into the doctor's office and you might be in a testing group. And some of the testing group would be given the actual medicine. Some of the testing group would be given a placebo, a fake medicine. And so they did this with a number of different pill types, which I'll describe in a minute. But in this case, it was an electromagnetic charge in the person's back to relieve back pain. Now, sometimes the patient got a real charge, and sometimes they got just a rumbling sound, but they thought it was a charge. And they looked at the groups over time, and they found immediately after the treatment, about 30% of the placebo group felt better. And that's okay. That's not bad. But 2 weeks later, that number dropped a little bit when they did follow-up studies of how your back pain was. And then by 2 months later, the placebo group had virtually no pain relief, whereas the other group, the group that was actually treated, still had a fairly large amount of pain relief. This is a classic example of a placebo effect study. Now, they've also tested the size of pills. Big pills are better than small pills. 2 pills that they give you, 2 placebos, are better than 1. If you paid a lot for your placebo, a high-cost pill is better than a low-cost pill. You believed it more. Capsules were better than pills. Shots were better than capsules. The placebo worked half as well as aspirin, and it worked half as well as morphine. Try to figure that one out. But in the end, these placebos do not cure the cause, and the results are fairly short-lived. And there's a great video on a couple of websites down below that I hope you find and read into it some more. Very fun. So personal experience can't always be trusted. In fact, your mind behaves differently to actual treatments, or whether or not you get the placebo, or whether or not you're non-responsive to a placebo. These might be skeptics. They just might not respond in the same way as the general public. So I'm asking you to just don't always trust your personal experiences as a way to gain knowledge. And now for some applications in ecosystem science. For example, in 1976, a long time ago, I believed that the accepted, commonly used line transect and point field methods used in plant community ecology were pretty good for studying the effects of grazing, fire effects, and monitoring vegetation change over time. I had read about those studies in textbooks and papers, and I sort of believed them. I hadn't tested them yet. So I wanted to move from belief to testing. Over time, new studies showed that the methods were somewhat biased, that they hit broadleaf plants, clovers and big flowers, but they missed narrowleaf plants, grasses, sedges. And they also missed patchily distributed species because we sampled along a line. And if a species was just off our transect, we would miss it. So there was a lot of error due to observation and where you happen to put the transect. And also the taxonomic training of the expert, so that any 2 technicians would get a different reading when we measured this meadow in Kings Canyon National Park. When I thought about it a little more, a little later, if I had to do it over again, I would have put in a series of plots, larger areas, and looked at plant diversity in these plots. I'd give equal opportunity for a narrow leaf plant and a broadleaf plant, or a tree or a shrub, to show up in these plots scattered across the meadow. Not in a line, because I want to capture patchily distributed species and perhaps rare portions of the ecosystem. In this other study, down in the California chaparral, I believe that environmental sensors provided accurate information on which to make management decisions. We could carefully measure temperature and relative humidity, and those readings would help us doing prescribed burns in the chaparral. Well, then I burned down the control plot in a study. I was quite proud of myself. And it sort of reminded me that maybe we just can't trust a single measurement. Wind turned out to be very, very important. Slight changes in wind helped burn down our control plots, places that were supposed to remain unburned. In fact, let's look at that sampling design. We had one hill, and one area was for spring burn, and then we had a fall burn area, and that was close to the control plot. Well, we burned pretty well in the spring because things were moist all the way around it. And then in the fall, the wind picked up and it burned not only the fall burn area, but the control plot. But this design puzzled me later. We subsampled within the spring burn area and subsampled in the fall burn area. Those are pseudo-replicates. Our sample size is really one each, one spring burn and one fall burn. And then we would have had one control had I not burned it down. In looking back, we probably should have had a different sampling design where we tested multiple hills, where we randomly selected locations for plots, and then randomly selected the spring burn treatment and the fall burn treatments from among the whole group of plots. Now, these fires would have been much more difficult to control and put out, but it would have given us better information, I think, on the effectiveness of spring burns versus fall burns in the chaparral. And now our sample size is, up to 10 each instead of one, because we've randomly selected the plot locations and randomly selected the treatments. This is a much more powerful design, even if it's more difficult to pull off in the field. So over time, I learned quite a bit about plant diversity. I realized that simple vegetation maps, for example, were much more complicated when you went out on the ground. When you went out on the ground, you saw ecotones and core areas of a vegetation type and gradients between the vegetation types. You had rare types, riparian zones and wetlands that were sometimes missed in the larger vegetation plots. You had elevation gradients and moisture gradients throughout the study area. So vegetation is far more complex than our simple designs allowed us to measure. And by simple designs in the grassland, we might have a tiny little Parker loop where we measure cover of species along a line. Or we put in Daubenmeier frames or other quadrats to look at vegetation, usually again on a line. And over time I learned that larger multi-scale plots were needed to really describe the vegetation. So we switched techniques by testing things. It ended up in a textbook. Great. But really, the story here is about curiosity, observation, hypothesis testing and analysis, and accepting or rejecting hypothesis before going back and learning from your mistakes and changing your designs, testing things over and over again. And if you choose a career in ecosystem science, you're choosing a wonderful career to develop curiosity, to make observations, to test hypotheses, to do your own analyses, and to accept or reject hypotheses, to learn yourself this way, gathering information, knowledge-based information. And it's a very fun career. Trust me on that. Thanks.